Hey y'all, welcome back to my channel. My name is Jeffrey. In this video, I'm going to talk about purine synthesis as well as the purine degradation slash recycling pathway. Let's get started. Purines are going to need two precursors to be synthesized, and these precursors are going to donate your carbon and your nitrogen. And you're going to need either one amino acid um, of these three, or a tetrahydrofolic acid, THF. Okay, so for your amino acids, I try to remember it as AGG. For me, that's a little bit easier to remember than GAG or GGA or any of the other combinations. So it's going to be glycine, aspartate, and glutamine. You just need one of those three to synthesize your purines. And for your tetrahydrofolic acid, you're going to receive it and you're going to modify it to become tetrahydrofolate in your diet, right? So you're going to need vitamin B9 or folate, and that's going to be converted into folic acid in your stomach and your gut. And through a series of reduction reactions, you're going to synthesize tetrahydrofolic acid. And you're going to do so with dihydrofolate reductase. And it's going to be the exact same enzyme for both of these pathways, and it's going to make folic acid into dihydrofolic acid into tetrahydrofolic acid. And it's going to in humans and eukaryotes, it's going to be inhibited by this drug called methotrexate. It's an anti-cancer drug. And you might imagine why it might be anti-cancer, right? Because purines are going to be nucleotides, and nucleotides are typically used in your RNA transcription translation as well as your DNA replication. And the only cells that really go through DNA replication are going to be your epithelial cells as well as your cancer cells. So this is the reason why we prescribe methotrexate for people with cancer, rapidly dividing cancer or metastatic cancer. Okay, the orange lines are going to be with your bacteria. Okay, so your bacteria can de novo synthesize folic acid from PABA. They don't need to have any vitamin B9 or folate, and they synthesize PABA through a series of reactions, and PABA is going to be your para-amino benzoic acid. And it's going to be inhibited, that synthesis is going to be inhibited by sulfonamide or sulfa drugs. So that's just another type of antibacterial that's going to prevent your bacteria from replicating. And another area of regulation is going to be trimethylprim. And this is going to affect only the bacterial version, the bacterial isomer of dihydrofolate reductase. Okay, so those are just some of the pathways that are going to be involved in your precursor synthesis. All right. And now that we have the two precursors, we need to synthesize our purines. And the base for our purine is going to be inosine monophosphate or IMP. You're going to synthesize that through, first, you have a ribose phosphate. You're going to use PRPP synthetase to add on a phosphate, another phosphate group to that ribose phosphate to make it into phosphoribosol pyrophosphate or PRPP. And the second that you make this compound, it's actually going to activate your phosphoribosol aminotransferase, which is really great because that's the next step in the pathway anyways. And it's going to take in a glutamine and it's going to make phosphoribosolamine. Okay, and phosphoribosol aminotransferase is going to be the rate limiting step in this series of reactions. Once you make phosphoribosylamine, it's going to be readily converted into your inosine monophosphate. And that's going to be your parent nucleotide. And remember, IMP doesn't really sound like GTP, GDP, any of our bases that we know and love. The base that it's going to have is hypoxanthine. Okay, so this is going to be kind of the parent nucleotide in which we can differentiate it into our different purines. Okay, so with IMP, we're going to use IMP dehydrogenase. And to synthesize adenosine monophosphate, we're going to use GTP. And to synthesize guanosine monophosphate, we're going to use ATP. Okay, and that kind of makes sense, and I'll try to explain it a little bit later. You're going to phosphorylate this AMP all the way down to either ATP or ADP. And remember, ADP just means adenosine diphosphate, and ATP is adenosine triphosphate, right? Because this was mono, di, and tri, okay? And we're going to do that for both. Now, when it is in the ADP and GDP level, when it has two phosphate groups on it, that's what we consider to be a ribonucleotide. And in this case, we can actually use this enzyme called ribonucleotide reductase to reduce it into the DNA form, right? The deoxy ADP and the deoxy GDP. And we can use that then in DNA synthesis. So for our S phase of our cell cycle. Okay, and these enzymes are going to be inhibited by hydroxyurea, and that's going to inhibit any type of DNA synthesis, right? And that's good for if we're trying to stop the rapidly dividing cells. And ribonucleotide reductase is also going to use these cofactors, thioredoxin, as well as NADPH. All right, and here's another different pathway. So for our feed-forward pathway, we have ATP that is going to act as that cofactor, or for IMP to GMP synthesis. So, and this kind of makes sense, right? Because we want equal amounts of purines, just as the same as we want equal amounts of pyrimidines. Because remember, Chargaff's rule, we have equal levels of A and G, and we have equal levels of C and T, right? So, yeah, so you have your ATP that's going to act as that cofactor for your IMP to GMP, and then your GTP is going to do the same for AMP. And your GMP synthesis can actually be inhibited by an enzyme called mycophenolic acid, and that's going to inhibit IMP dehydrogenase for that GMP pathway only. All right. And then once you synthesize your ATP and your GTP, if you have excess amounts, this is going to act as inhibition of your PRPP synthetase, as well as your phosphoribosome aminotransferase. And that makes sense because remember, if you have high levels of ATP and GDP, that means you also have high levels of your ribonucleotides as well as your deoxyribonucleotides. And if you have high levels of those, you obviously have a lot of purines and you have overabundance of your purines. So you're going to stop these two critical enzymes in making your purines, okay? And then if you have any leftover deoxy ATP, GDP, deoxy ADP or deoxy GDP is going to eventually go through turnover, as well as when your cell dies, you have those extra nucleotides around. So obviously, we've spent a lot of energy and we've spent, remember in this pathway, we've used a lot of GDP, ADP, we've used a lot of kinases to transfer ATP, phosphate groups from ATP and GDP. We've used ATP many different times in this pathway to help synthesize these purines. So it's very energy expensive, right? We want to keep those around if we've already synthesized them. So most of those purines that we've made, we're going to go through purine recycling, and then some of them we're going to degrade. So roughly 20% we degrade into uric acid. It's going to go through a series of pathways and eventually lead to uric acid. For purine recycling, there's going to be two different enzymes that mediate this process for purines. And the general pathway is just going to take the free purine base and it's going to go through a reaction to make a purine nucleotide. So when we look at APRT, which is adenine phosphoribosyltransferase transferase, you can kind of infer that it's going to do something to adenine, right? It's going to transfer a phosphate group onto the ribose sugar, okay? So it's going to take the adenine and it's going to take PRPP and it's going to add a phosphate group and make it into um, adenosine monophosphate, right? AMP. And then for HGPRT, we're going to have it's going to be called hypoxanthine guanine phosphoribosyltransferase, transferase. And that makes sense because you're going to take hypoxanthine and you're going to take guanine. It works for both, and it's going to add a phosphate group onto the ribose sugar. So it's going to go through this pathway with PRPP, and it's going to synthesize your IMP, which remember has the hypoxanthine on it, and then the GMP as well. And then just a side note, Leishman syndrome, which is a X-linked recessive disorder, um, it's going to inhibit this HGPRT enzyme. Okay, so you're not going to be making any functional copies of the HGPRT enzyme, and as a result, obviously going to you're obviously going to have a large buildup of your hypoxanthine and your guanine, so you're going to have this buildup of purine bases. And eventually, obviously, if it can't be recycled, it's going to be degraded. So then eventually you're going to end up with a ton of uric acid in your body, and this is going to be very toxic, especially in your brain. So typically, these patients have many different symptoms, including self-mutilation as well as intellectual impairment. So I hope that this video has been helpful, and I hope you have a great day.